Hello, I'm Elijah, and today I'll be showing you the Amnis machine, which is also known as the imaging flow cytometer. So a quick summary of what this machine does is that it combines the functions of both a flow cytometer and also a microscope. And what this does, this machine is able to acquire fluorescence images and also acquire the shapes and the pictures of cells that you are interested in looking at. And for my project, what I've used this Amnis for is for signaling transduction, and in particular, I'm looking at the nuclear translocation of transcription factors. And on this window, you see an example of analysis of the nuclear translocation experiments. So what we have is that normally we'll have an image for the nucleus and also an image for the fluorescence that detects the transcription factor of interest. And then they also have that emerge image that you can see when the transcription factor actually goes into the nucleus. And below you see a few graphs where we do the gating, and we do, not we, but the computer actually does the algorithms and the analysis for us to determine whether translocation has occurred or not. Right, so this screen here shows the software itself and what we see during the acquisition of the data. So we do have the various channels. We have up to 6 channels here, and one of which is we used for the bright field image, another we use for the side scatter, and the remaining channels we can use it for the various fluorescence. And in our case of analyzing nuclear translocation, we normally use one channel for the nuclear stain and one for the fluorescence of the transcription factor of interest. And during the acquisition, you can see the various images of the different cell events that we acquire. And towards the right, we see the dot plots of the acquired images of the acquired events. And from there, we can do the gating, where we start to see the events of interest and the parameters that we are interested in looking at. For example, the cells that have been translocated into the nucleus. And just like a flow cytometer, we can set the acquisition events, the number we want to acquire, and also the gating that we are going to set as the limit to set the maximum threshold of the cell events they want to acquire. And also, one thing that we can adjust is also the magnification, where difference from the flow cytometer is that we can determine the magnification that we would see the cells at. So this is the part where the image acquisition similar to a microscope comes into play here. So in my experience with using the Amnis, I would say that it's a pretty interesting machine, considering that it has both the capacities of a flow cytometer and also a microscope, which enables us to acquire images. Above all, it's able to do some pretty high-intensive data analysis, which involves the use of various algorithms. And it enables us to do certain experiments which we couldn't have done it with the normal flow cytometer. And in this case, the Amnis itself enables us to do a high throughput screening and also high content imaging as well. So above all, around, I would say it's a pretty unique piece of equipment which will be suitable for use. And one particular interesting aspect is that it can return the samples that you've collected. So if you're worried, you could actually collect the samples and run it again on the flow